Hi, everyone. Welcome to the last panel of the day. Hopefully we can keep you awake. I'm super excited because we have a bunch of CEOs that have been leading the way across many different CMTs, as well as using different modalities. And I guess my hope is to give you all some insight, or have them give you some insight into why we pick indications, why we pick CMT, and maybe some sense of the day-to-day struggles that test our conviction for the targets and indications that we go after. And I'm going to kind of skip over intros and go straight to the first question for the panel. And I've actually spent probably the last 2 weeks, like five days, sorting through targets and trying to help companies pick indications and targets, and whether it's art or science to pick an indication. So the first question I want to ask, and we can just go across, is what's your general approach to selecting targets and indications, and what has led you to choose CMT1A or CMT as one of the indications that you're going after? So we'll start with Gerhard and go down.
Yes. Good afternoon or good evening. My name is Gerhard Koenig. I'm not the CEO. I'm the executive chair of Augustine. But one of our team members, the tall guy over there, he gave a talk earlier this morning. I know his data. They're very good. So just a few words again, sort of as a cliff note on Augustine, HDAC6 inhibitors, small molecules, proximal, and then leading into an answer, very proximal biomarkers. I hate HDAC for the word of it because it's not a histone deacetylase, it's actually a tubulin acetylase, as you know, inhibitor, our compound. So first of all, it is that you affect the microtubules, the highways of a neuron. Neurons are very long, as you know, and that really attracted me when I learned from the team, the science, that you have a very proximal biomarker, you inhibit the enzyme, you can measure that in plasma. Translatability is huge in our field because we have so many. Tim actually and I, we have worked before together, and we know about failure also, and he can talk about that too. It's often in PNS or CNS that you have good targets, you can maybe track the targets, but you lack suddenly your beautiful animal data. We all know we can be cynical about animal data in PNS or CNS. And it's the biomarkers, which you heard a lot about. We have, not just us, we have beautiful biomarkers which correlate with the animal behavior, with functional endpoints. So that's an important aspect for us, for CMT. So sort of at face value, it's all coming together. Functional, biomarker, which you can measure almost with the same methodologies in human plasma, not just in animal plasma. So we have those data, we have not just animal behavior, we have also functional velocity and so on and so forth. So this is a composite of data, which really, when I looked at it, is like, I've not seen anything better actually in my career. And so I felt that's actually a strong point for us with this to prioritize CMT, and we're also very happy that we see the momentum in the field. One thing, you haven't asked that yet, but I am concerned a little bit about the endpoints, the lack of progression. That's something which I'm thinking a lot about now, that I'm learning more. I started really getting more into the data about 3 months ago or so. And when I hear how we approach clinical development, I think that's something we need to talk more about, not just biomarker, but beyond that. How can we convince the regulatory authorities that the treatments do everything they're supposed to do, but we need to do a 5-year trial to see that? So just being a little bit provocative, right? The numbers we need, the statistics we need, that's a big concern for me right now. So I don't have a solution for that yet. So
we're going to talk more about the challenges with clinical development and investors in a little bit. So we'll probe into that. But Tim, do you want to take it away?
Hello, Tim Piser. I'm actually also not a CEO, head of R&D at Samsara. And at Samsara, we're developing small molecules for the treatment of neurodegenerative disorders. We follow the biology, and so we came at these compounds by testing them in assays for the activation of autophagy. But when we use the compounds, not only to optimize them as small molecules, potential therapeutics showing efficacy in animal models of CMT1A, for example, very encouraging. We also were using them to interrogate their mechanism of action. Autophagy is a very complex biology. There are many mechanisms involved in the initiation, propagation, and proteolytic degradation through autophagy. And what we found was a linkage to PMP22. The complex in the ER that these compounds appeared to stabilize in limited proteolysis mass spec included PMP22 interacting proteins. So that immediately oriented us to PMP22 and to the animal model testing that we've done. So it's interesting that you start with a platform with complex biology, you find small molecules, and those small molecules lead you to different targets, actually. And we found some selectivity even further than that. We have lead programs in ALS and Parkinson's, and the compounds that we are finding modulate PMP22 don't necessarily work in ALS, iPSC model, motor neurons for TDP43, for example, in ALS, or for synuclein and patient-derived iPSCs of dopaminergic neurons in synuclein pathology and Parkinson's, and vice versa. So the small molecules find their targets, and then you find actually there's quite a bit of selectivity that maybe direct you to target a particular indication, in this case CMT. The opposite is also true. Once you discover a new mechanism, you may find a broader therapeutic application than you had thought you might have initially. And I was pointing out from the podium at the Blitz presentation yesterday, our poster's out there, number 7. Folks have recently pointed out that some of the Vertex small molecule chaperones, if you will, pharmacological chaperones for CFTR, actually also modulate a CFTR PRAF2 interaction. PRAF2 is one of the proteins that interacts with PMP22 is in this ER complex. So maybe there's a broader therapeutic application beyond CMT for these compounds, but certainly within our immediate focus, CMT is our lead indication. So it's just following the biology to get to the indication that you're prioritizing.
Good afternoon, everyone. I'm Rachel Salzman. I am the CEO of Armatus Bio. Anyhow, so for us, what we're doing at Armatus is we're using vectorized RNAi to address neuro and neuromuscular diseases. So that means if there's a disease that's autosomal dominant, so in other words, there's a gene that if it gets expressed, it's toxic, and as a result of it being toxic, it causes neuromuscular phenotype in people. That's what our technology does. You kind of got a prelude to some of the biodistribution that Dr. Scott Harper presented earlier today. So when you think about pointing that platform towards a disease that is autosomal dominant and is neurological, CMT1A is just, here I am, because we know what the cause of CMT1A is. It is an extra copy of PMP22. We know that if there's too much PMP22 protein, it is toxic to the myelin and you end up with a peripheral neuropathy, although I will certainly agree that I'm not sure that we understand all of the mechanisms of that pathophysiology, but we do know what the etiology is. And because our therapeutic platform directly targets that overexpressive toxic cause of CMT1A, for us, it's kind of an like I said, it's an in-your-face indication, because the one part that maybe we'll have time to get to, and I think Artie thinks about it a lot because he's been through it, is there also has to be a business case. And in this case, the business case is simply not just that we know what the disease is and we understand our technology and how we're targeting it, but it's a very frequent disease for an orphan indication. So we really believe that the very prevalent, serious unmet medical need is quite compelling as well. Thanks.
Thank you, Artie. I'm Ilan Ganot. I am also CEO. I'm also the only guy on the panel without a doctorate degree. That's great. And I didn't choose CMT. I guess it chose me. Before I was chosen by Duchenne muscular dystrophy. And after doing that for 10 years and taking a bit of a break, I got a call from some guys in Europe that had a really neat mechanism that they were working on in cancer. an integrated stress response related mechanism to, shrink tumors. It looked pretty interesting. But then they came across Rob Burgess and some work he did in CMT 2D, and thought, wow, here's a total unmet need with enough patients to make it worthwhile to really try to go and see if we can make a difference. And they kind of gave me a call, and that's where we are. So we are indeed trying to identify whether there are other tRNA synthesizers that we can be helping with that ISR pathway. There's not a ton of models for many of those diseases. I had a little bit of time with Max just outside now, which is an example of one of those tRNA synthesizers that don't have a model, but we definitely understand the mechanism and we assume it may be related, but we need to figure out how to prove it. One way to do it is in patients, but it's not easy, cheap, or quick. Ideally, there'll be better models to do it with, and so we're trying, and we're working with actually some, groups that Max helped connect us with. And at this point, we are quite optimistic that we can work with 2D and 2N, and hopefully more soon. So a little bit like you said earlier, the pathway is the pathway, but the target community you could be able to work with may be growing as we improve our ability to understand. Thank you.
David Goldstein, also on the CEO side of the table here. I do have a PhD, but it's so far away from most of what we're talking about. I think we're in the same boat, actually. The way we think about targets is really picking targets where we can be completely confident of the biology before we get into the clinic. And I have to explain a little bit about what I mean by that, because very few drug developers are going to get up and say, yeah, we're working on this target, but we have no idea whether or not the biology is right. So you're generally going to say that you believe in the biology. However, it really is the case that there are many circumstances where the biology that you're focused on really is a hypothesis that can really only be fundamentally resolved when you get into the clinic. And really the illustration of that is where you have a biological process and you've taken the view that that biological process has a lot to do with how disease is actually caused. And you might, for example, if you're talking about genetic conditions, you might say this particular genetic condition, well, the way you actually get disease has a lot to do with autophagy, or it has a lot to do with inflammation, or has a lot to do with something else. And then you actually go after that biological process in order to modulate the symptoms of the disease. But you can't really be certain that you know quantitatively how important that process is to the disease until you get into the clinic. Now, before I go on to say what we do, I want to start by saying that way of working in drug development is an important, critically important way of working. And so by focusing on what we do, I mean no criticism of that approach to drug development. It has to be pursued because there are many things where you can't really know until you get into the clinic, and we need inspired biological hypotheses that are then clinically tested. We, however, work in a different way. The way that we work is really by picking targets where we can be genuinely as confident as you can possibly be that we have the biology right. And so what that means is we work on targets where the clinical population we're going to work with is completely homogeneous. So we know exactly why every patient has disease because of a mutation in an individual target. And moreover, we know what those mutations do to cause disease, and they have consistent effects, all the different mutations in the target. And so if we really do know all that and we're right about it, and it's really hard to be right about those things in targets, as we discussed earlier, then if you can actually go and undo the underlying cause of disease, and it's the same underlying cause of disease in all patients, you really do have the biology right. And so we've actually worked really, really hard to pick out those opportunities. And the reason that we have is that it's been a real frustration of mine to be looking at these targets that are responsible for devastating diseases and take the view, you know what, this is completely doable. We actually really have the possibility of pursuing a predictable drug development program that can undo the underlying cause of disease. And for all of those things, we as a community, I believe, have an obligation to make sure we do that work, do it as fast as it possibly can be done, because we're going to end up with an important therapy predictably. The target that we work on within CMT, TRPV4, is like that. As I mentioned earlier, the mutations cause disease by activating the channel, and you can actually develop a small molecule program that undoes the effects of those mutations, and it really means that you have the biology right. So that's the way that we pick targets. And just to really emphasize, it is absolutely not the only good way to develop treatments, but it's the way that we do it in the company.
Thanks, everyone. So now I'm going to ask you maybe some thorny rapid-fire questions. Hopefully we can keep the answers a little bit concise so that we could get to more material. But what I'm trying to do here is have the audience appreciate some of the questions that we get and the challenges we have as not only drug development, but the business of drug development. And so maybe the first question is geared towards Gerhard and Tim, as you guys work on targets that probably have applications across multiple indications. And so it's okay if you don't work on just CMT, but take us through, an elevator pitch on, what would lead you to pick CMT? What are some of the challenges you face as you pick the indication, in committing to CMT or the other target?
Yeah, so that's not thorny, or whatever you call it. That's a good question, because it's actually happening right now with us. Like, I'm also CEO for another company, so I know about fundraising, Arcuda, and I'm coming to that in a moment. Arcuda worked on FTD, frontotemporal lobe dementia. We started out with ghrelin subtype. I have a lot of background in Alzheimer. I worked with Tim on schizophrenia, so I've experienced in various fields. And coming back to fundraising, I can tell you that investors do like CMT. That's my experience so far. When I talk to the team, they do like it. They find it plausible. At least that's my preliminary assessment. Now, obviously, our approach with small molecule, right? Ease of use. We have, for example, beautiful data. I don't know, actually, did you show it, Frederick? Yeah, on CIPN, chemotherapy-induced polyneuropathy. I mean, the data are beautiful, right? And the biomarker and everything works out, and you can, David, there's a plausible story that there's translatability, that it would work. Interestingly, investors don't like that so much. I can tell you that one. It's not that they poo-poo it, but they like that we focus on CMT. Some of our corporate investors think different about this, so it's not such a big tension. I cannot tell you how it's gonna work out in two and a half years from now when we're there, so that will be a new story. But I'm familiar with this. You just have to be open about, if somebody wants to do a bigger trial and a bigger indication, you need more money, right? So show me the money, right? So I'll stop there.
Tim, do you have anything to add?
So I really, the only person who's not a CEO today, just to be clear.
I did learn a lot from him.
Help CEOs address these challenges.
I'll just repeat. These small molecules we're pursuing modulate PMP22 proteostasis. They decrease it in vitro and in animal models of disease, and that's the cause of the disease is too much PMP22. So straightforward reason to focus on CMT.
So maybe to another thing that comes up a lot is prevalence, right? David and Ilan, you're working on CMTs that are certainly not 1A and have 75,000 patients in the US. Can you talk about some of the challenges with prevalence and maybe even take us through how you get investors excited about, or pharma excited about what you're doing, given how rare it is?
I think prevalence is, on the one hand, overrated, and on the other hand, well offset by orphan drug pathways that exist exactly in order to create incentives in the system to have people work on diseases that may not, without some help, be commercially interesting. I cut my teeth in this business observing an interesting phenomenon of how a drug for a small part of a rare disease, in this particular case a specific exon skip in Duchenne muscular dystrophy, that probably doesn't work, was worth billions. And it may not be the best example in terms of, because I wish the drug worked better for those patients, for sure, but it attracted investors. There were enough patients to try the drug in, and once it was approved, it was a commercial success. So I think there are great ways to elevate the patient number problem and to manage it in a, in a drug development campaign. I mean, the super ultra rare, when you have 5 patients or that you can't even find, those I think are more difficult. But if you're in the tens to hundreds in the US, I think there's already enough to talk about what can be done here.
So if you're asking us to be a little concise, and I think my concise answer to the question, how do you excite investors for the rare diseases, is I don't know. But the slightly amplified answer is I really do think that it's very hard to predict how much money a medicine is going to make. It's just, everybody tries to predict that. This is basically always wrong. But one thing that is, I think, fair to say is that the experience is really good drugs do well. If you actually look at the history, if it's a really effective drug, it ends up doing really well. The estimates of prevalence are often wildly wrong, but really, really good drugs end up doing well. And the other comment to make, amplifying on Ilan's comment, for the 4,000 odd genes that cause Mendelian diseases, even the ultra rare ones, it's very rare that there's only a handful of patients. The way that the mutation opportunity works, it's usually that they're in the hundreds. And for most of these conditions, therefore, there probably are enough patients out there to allow you to run trials, to allow you to have some market.
So Rachel, we have a lot in common when, 1A, obviously, but when I started DTX, RNA therapeutics were kind of out of vogue. No one cared about them. They killed everyone, according to, and sort of we heard a lot about gene therapy today and its promise, but there are real challenges with raising money in gene therapy, despite some of the most promising primate data that's at least in the public domain right now. Can you speak to maybe the more difficult side of gene therapy and raising capital, and specifically related to some of the challenges you face for CMT1A and overcome it? I know that's a lot, so maybe 2 or 3 minutes, but I think it's important given how much time we spent on gene therapy today.
Yeah, Artie, I think it's 2 parts. I think one is a little bit agnostic to the 1A program because the entire field is feeling a lot of pain when it comes to fundraising. If you have, I'm going to say, one-time genetic medicines, we can call them gene therapies. I think Scott defined it earlier. Let me just first talk about that, and then I can talk about our way of overcoming it or some of our strategies. So part of the issue is a lot of these drugs actually really do work, and they're not having great launches, okay? Now, it's early days. I think the Celia from FDA put up the list of drugs, and if you looked at the years they got approved, it was relatively recently, but they're not having these amazing commercial launches that the companies themselves had guided. If you look at BioMarin's hemophilia drug, they said it was going to make a lot more money than it's making now. And then, I'm just giving a random example. I'm not pointing a finger at BioMarin. I could name 4 other companies. So the point is simply this. We have a situation where I would say clinical data is actually great, and the drugs are not having smooth, highly successful launches. So the challenge there is anytime somebody gives you $1, all they're thinking about is how am I going to get out of this, and how am I going to get out of this with more than $1, right? So what is really important to understand is from an investor's perspective, they can see the present and the future at the same time. They see the present, they see our data or another company's data, and then they see the future because they're looking at a gene therapy company that actually developed a successful drug and got it approved and is not making money and is trading at a very low valuation. So the challenge there is they're saying, these are great data, but I don't see how I'm going to get out of this. It's called an exit, right? Which is either going to be the company goes public or there's going to be M&A. Your M&A was a great benchmark. Okay, so that's sort of the generic problem right now with gene therapies. In terms of how Armatus is taking, I'm going to say, a differentiated approach, we're able to position what we're doing as favored market dynamics. We believe, based on what I explained earlier, that we have so many patients and that you saw we can get the kind of biodistribution with a relatively modest dose of a drug that is generally expensive to manufacture, but the dosing that you give for something intrathecally is about a log lower than something that you give systemically. So the point here is we're building a target product profile of a drug that is transformative, addresses an unmet medical need, and because of the parameters of the manufacturing that are unique to the kind of biodistribution that we're looking for in CMT1A, we can actually go out there at a price that is a sustainable price that is reimbursable. So in other words, we can have a successful launch. And when you look at those other companies, they either have very, very rare populations or there's competing standards of care, like in hemophilia, that patients are already happy. CMT1A patients do not have an approved medicine where they're happy already. So these are some of the strategies that we're taking.
Awesome, and thank you. So let's, slightly switch gears a little bit. Gerhard brought this up earlier. At least in my experience, you have this great preclinical data. We saw tons of it today. But that's sort of just the beginning when it comes to the business of drug development. And so, when I talk to investors, a lot of the time was about clinical path and milestones or checkpoints along the way that are going to guide you, to know if you're on the right track or not. So maybe this will be a whole panel, but we can start with David, because I believe that he's on the verge of, or his company is on the verge of maybe traversing this in CMT in the not so distant future. What are the kinds of questions you get about the clinical path, and what gives you conviction that you can answer the acute questions around the acute activity that will drive investment in a longer trial?
So the challenge is, of course, that we don't have endpoints that have a lot of precedent around them in CMT, and I think it's a real challenge because you have a lot of comfort when you're using well-established endpoints that that the regulators are comfortable with, and I think that makes it more difficult. And I will say that although there is some growing enthusiasm for CMT, I think that the absence of well-established regulatory endpoints has been a major constraint on the field, and I would say that CMT is getting now much less attention in industry than it should be getting for exactly that reason, and it's going to dramatically change when there are a few approvals that come through and people get comfortable that you can have endpoints that lead to approvals. And one of the problems in especially pharma, but biotech world too, is that there's this phenomenal herd mentality and aspect, and everybody kind of moves in one direction at the same time, and there has been sentiment against CMT because of challenges of accepted regulatory endpoints. So that's an issue for us. It's an issue in CMT overall, and it's an issue in particular for us because we're working in a particularly rare form of CMT where we don't understand the disease presentation all that well, and lots of people are working hard to better understand it and try to figure out what are the most important features of the disease to patients and what would constitute an appropriate regulatory endpoint in terms of what's important, and a practical endpoint for trials that you can actually measure consistently and accurately, regardless of the severity state, which is very, very variable in patients, and that has a reasonable chance of actually moving over the time frames that you can actually look at patients in the context of a trial. A lot of those things are questions right now. I can say, though, that I myself derive a lot of confidence from what we see in the really terrific preclinical work that I talked about earlier, where we really see a dramatic impact of therapy after symptoms emerge. And so the way that I really look at it is, even though we don't have a completely reassuring answer that I'm going to use this very well-established regulatory endpoint, I believe that we're going to have an important impact on the presentation. And if we have an important impact on the presentation, that will ultimately lead to approval. It might take a little bit longer than we would hope if we don't pick the right one initially. But if we have an important impact on the presentation, it will ultimately lead to approval.
Anyone else want to add anything?
Maybe something a little bit orthogonal to this is, as I mentioned before, have been working in frontotemporal lobe dementia, obviously in Alzheimer's, schizophrenia. So what is the issue with schizophrenia? When you're schizophrenic, you're not really in front of anybody anymore, right? You are blocked away. What does, Parkinson's disease have? They have Michael J. Fox, right? We all know much more. Very good foundation. I worked with them. Very good foundation. So what we need in CMT, and maybe I'm saying something ignorant here, but I'll just try to be provocative before we drink. We need somebody famous. We need famous people. I worked in FTD. I know lots of, and this doesn't mean to, negate the suffering and the severity of anybody who's not famous. But the reality is, Bruce Willis has FTD, and suddenly people talk to me. So if we have in CMT, which is actually a big disease, right? Some people argue and have told me it's not even orphan anymore, totality of it. If we have one or two celebrities who are willing to talk about their disease, the regulators will work much better with you. Because we've seen in ALS what that can do. And I don't mean this in a threatening way, that's just my intensity, the way I talk. But I feel this is something I've thought about. What I've learned in a few months? I work more intensely with the company. I feel it's an underappreciated, long-term, severe, but not immediately lethal disease. So we need to have somebody talking about it. And go on Capitol Hill, wherever you go, and make this a cause. I live on a COMAF. I mean, I have every second week a walk for cancer, and there's nothing wrong with it. Who walks for mental health or diseases like this, right? So turning it around a little bit, and this is not a critique, but it's just like, how can we be more effective? Because if we don't have good clinical endpoints, we have to rely on biomarker, on projections, and then convincing if our treatment is promising, doesn't have the full efficacy package yet, but the right biomarker moves. David is going to present how it's basically absolutely clear, convincing. You might have a better chance getting approved. Just a thought.
So maybe you led me to this. I mean, I have to ask this question, but maybe what, can the community, whether it's the CMTRF, whether it's the INC, are we doing enough to support you in terms of gathering natural history data, looking at biomarker data? Is that work helpful? Are we doing enough? And if not, what can we all do differently to enable you to move faster?
No, not doing enough.
So expand.
I mean, we really should have really systematic natural history studies going on for all of the forms of CMT. They're different from one another. We know very little about a whole bunch of them. We need really systematic natural history studies and really systematic biomarker studies. That doesn't exist. So that would really ease the path for drug development.
And you talk to other foundations. The FTD group has benefited tremendously from the Diane group, Alzheimer, because Alzheimer, as you know, went through, a phase of depression because nothing works, and there was a lot of critique. A lot of investigators in the Alzheimer community moved from the dominant inherited into the FTD field because there were gene mutations. Granulin is one where we've specifically focused, and they did a great job quickly getting up to speed about the natural history. If you link up with them, they will help you how to do it logistically, what to do, have a program manager and so on. Within 2 years, you can do a lot. And that helps you then eventually to supplement maybe not the most efficacious trials yet after 12 or 18 months. Because I'm getting a little bit nervous if I think about 3 or the patient populations we need to have a pivotal trial. So anyhow, I'll stop there. So
I'd like to make a very specific suggestion, tying together a couple of things that we've heard. So in ALS, we've heard the story about neurofilament light chain, how it transformed the development of tofersen, the linkage to the clinical outcomes. And now, and if you go back to tofersen, in animal model days, you saw a reduction in SOD1 expression, and you saw a reduction in NFL in the animal models. And now in ALS, that's what everyone wants. They want to prolong survival, reduce neurofilament light chain in animal models. We have compounds that do that. That's why we're excited about them in ALS. In CMT, you have clinically the muscle fat fractions repeatedly showing really sensitive measures of disease progression. We heard that earlier today. In neuromuscular disorders, in mice models, you can measure fat fraction. You can measure muscle atrophy in those. I've never seen anyone measure that in a CMT model. If someone has, tell me about it. And if they haven't, you guys should do that. That's your strongest endpoint. That's possibly predictive of treatment outcomes. It can be applied in your animal models. A lot of really good preclinical work using endpoints around neuromuscular performance and nerve conduction velocity, but those are slow and insensitive endpoints clinically. Use this muscle fat fraction endpoint in your animal models.
we had a discussion the other day just about that. I agree with you. That's something which one get it tighter a little bit and get the data. And that's something maybe you don't have to do just in your company, in terms of animal. You can maybe work with other companies as well. So it's a good point.
If you're going to use MRI, it's going to take an investment, and it'd be great to have access to that kind of capability out there if it was available.
The only thing I want to add to this is wearable and sensors. I think the technology, that the way it progressed in the last decade, things we can do now we couldn't do 5 years ago, not even close. I have personal experience with a drug that was able to demonstrate a statistically significant change in 3 treated patients against themselves untreated in a gene therapy. Not particularly great gene therapy, but it was the sensitivity of the sensors and their ability to measure the change was something you could never do with the naked eye. No matter how many doctors stand with stoppers, they will never, and in any regulatory interaction that I have been a part to, the regulators asked for more of that.
I've seen something like this in Parkinson's, where they have actually, basically the patient, is daily activity, how often they walk, how many steps they do in the house, how long it takes to go to the fridge or whatever they have to do. And they were able to, basically with computer models, very easily say, look at the improvement percentage of this individual.
But also you cannot cheat, because the problem we have with so many of these functional outcome measures is that, Jerry Mendel put out a paper about how kids would walk longer than the 6-minute walk if you give them $50 at the end, right? So you cannot cheat something that you're constantly wearing for 2 weeks. It's not effort driven anymore. It takes out a big chunk of the questionability around that outcome.
Artie, we saw a presentation at the Blitz the other night from Kayla at University of Sydney. She's probably here somewhere. Yes, she was showing the biosensors, right, from Biosciences, measuring really sensitive measures of movement deficits, basically in patients. These could be transformative. They're far more sensitive than clinical assessments. We took this approach previously in spinal cerebellar ataxia. I have no doubt it will work here, and you'll have more sensitive measures. I think it's a really important endpoint. That platform can also measure falls, really strong predictor. That in itself could be a registration endpoint, and patients don't have to report it. They just go home wearing the sensor, and it will tell you whether it happens or not. So I think it's a really important clinical trial readiness approach.
Tim, you stole my thunder because I was going to bring up her talk as probably my favorite talk so far. Of course, I was wanting more, like additional controls and everything, but it's a great start, and I don't know, we need more. I agree, we need more of that. So, man, we only have like 5 minutes. So, I mean, is the advice to have more intensive focus on sort of the next CMT up as we go think about natural history studies? Why is the current ongoing ACT CMT study not good enough to meet your current needs?
Just real quickly, because I think the gap was called out during the biomarkers and outcome measures. We don't have enough longitudinal, or I'm going to even say cross-sectional data that, I'm just saying what you all said already, as that the fat fraction, how that relates to function. And I don't think that's a heavy lift. I'm not even talking about animals, I'm talking about humans. So to me, that would be one thing. And then one other comment is, I know when I go up to anybody, I'm like, how are you? And everybody's fine. It would be helpful if everybody sort of wasn't so stoic, because I think sometimes when I go to speak to neurologists that this isn't an area that they're active in, what they remember from when they were taught about CMT is like, oh, it's no big deal. So that's why I really appreciated the plenary opening speaker who really talked about what a true burden this disease is.
Okay, so maybe I'll end with kind of a fun question. I think it's a fun question. Maybe you guys have had some experiences, but I guess in my experience at DTX, there was a lot of dogmas out there propagated by investors, by KOLs, that we sort of discovered not to be true. They weren't backed by clinical data. They weren't backed by preclinical data. Are there any fun things that you've uncovered that you don't even have to say how you resolved it, but that are just, you don't believe to be true about CMT research or CMT clinical literature? I can name like 10.
I think we're about to learn, if I paid attention to the earlier presentation, that you can, in fact, have a positive clinical trial in this slowly progressing disease. That's going to lift everything. It'll change the game. So I think maybe that's the key thing. The key dogma that you hear from people who don't follow closely is that it's too slowly progressing to detect change, and you can't mount an effective clinical program. And I think one marketing authorization will change that discussion.
I mean, I was just going to add one kind of follow-up to that. I think there are data in other indications that, like, for example, on the electrophysiology, right? We saw data today. If you talk to, well, I won't call out any specific INC member, but if you talk to almost any of them, they will say, you can't bring back an action potential. You can't do it. And it's just not true. There's TTR polyneuropathy. Now we saw this morning where 0 SNAP, and then all of a sudden you have it. So anyway, David, sorry.
Yeah, well, you've both already said it, but it's so important, I'm going to say it again anyway. I mean, the biggest one really is this perception that all you can do is arrest progression. And we'd have, I think, absolutely definitive evidence that that's not true. That there are going to be lots of examples where we're going to be able to improve, and we shouldn't be shy about saying that's what we're trying to do.
And all the preclinical data, I'm mostly familiar with 1A, says you can take no action potential and bring it back in a really robust way. We've seen it in 5, 6 labs now, and if you just read the EMG report, you would say, well, that's never coming back, and you'd give up. And the mice are running around again, et cetera. So, that was one of the dogmas that I was hoping to get at. So I don't know if anyone has any final thoughts that they want to leave us with. Speak now. All right, well, thank you to members of the CEO panel that are CEOs. And those that filled in for their companies. I hope this was a good panel and you learned some things that you didn't know about the day-to-day struggles and the business of drug development and why we pick CMT.
Thank you.